Hi, here we are back for the audition lecture. So audition is your sense of hearing, and that's what's going to be the topic today. So show me my little highlights. All right, the first thing we're going to be talking about up there, point one, is what is sound and kind of how do we measure it? How do we think about it? So sound is a perception. Sound is a perception that's created by your body when it interacts with energy that's out in the world. In the same way that vision is a product of your photoreceptors in your retina interacting with light waves and sending that information, changing it into a neural code in the photoreceptors, sending it back to your brain for processing. Audition is the process of taking sound waves and translating them into the perception of sound. So what are sound waves? Sound waves are created when objects interact in the environment. When objects interact in the environment, maybe it's like your two hands coming together and clapping. When these objects interact in the environment, it creates energy that comes off of that interaction. That energy vibrates the air molecules going out from it. And as the air molecules vibrate, those vibrations travel in waves, and they can go into your ears, and they do go into your ears. And if you have a functional auditory system, they're changed by cells in your ears into neural energy, which is then perceived by you as various sounds. So, the question, if a tree falls in the forest and there's no one or nothing around to hear it, does it make a sound? Well, we have the answer to that question. No, it doesn't. What it creates is a series of vibrations of air molecules. That will happen even if there's no perceiver there. But there has to be a perceiver, a receiver, in order to take those molecules and translate that vibration of molecules into something meaningful for the organism. So we've answered that philosophical question. So how is sound measured? Well, there are two main ways that sound is measured. And one is called frequency or pitch. So frequency or pitch is what sound are you hearing? What note are you hearing? So imagine you're playing a piano and you play all the different keys. All the different keys are different notes. Those different notes are different notes because they create different frequencies. They vibrate the air molecules in the air at different sound waves. It's actually waves of energy traveling through the air in the air molecules. And those waves can travel very slowly, or those waves can travel very quickly, just like light waves. There can be short, medium, and long sound waves, just like there can be short, medium, and long light waves. Okay, so that is what tells you what note you're listening to, how your brain can discriminate between different notes. Well, that's all well and good for understanding something like music, but what about other sounds? They're made up of notes too. So all of the different sounds that you make with your voice box, you're moving air. Air is being sent up through your voice box and out of your mouth, and your mouth and your lips and your teeth and your voice box are shaping the air that comes out of your mouth, and those are different frequencies. So the different sounds in language, the reason they're different is because they're different frequencies. A frequency that's the letter B is a different frequency than the letter K. And that's a different frequency than the letter M. And that's a different frequency than the letter A. Okay, those are all different frequencies, and so you translate them in your brain as different sounds. So So the way that we talk about frequencies is in hertz, okay, hertz, H-E-R-T-Z. And hertz is also abbreviated HZ. You might see that in when we're talking about different frequencies. Around 4,000 hertz is what we usually hear. What does that mean? Well, 1 hertz is 1 wave cycle in the air per second. So if you had 1 air molecule wave cycle from the beginning to the top to the bottom and then back to the middle, that 1 full wave, if it took 1 second for that wave to make a full wave, that would be 1 hertz. So 1 wave cycle per second is 1 hertz. So 4,000 hertz is about the middle of human language, and so that's 4,000 cycles of the wave per second. Humans start to be able to, our ears start to be able to pick up sound, depending on how sensitive your particular equipment is, between 1 to 500 hertz. So, or 100 to 500 hertz. So if something is under 100 hertz, people just can't hear it at all. It still exists. If something is 50 hertz or 25 hertz, it still exists, but we can't pick it up with our equipment. But there are other animals that can. So for example, whales, big whales send out their whale song at very low hertz. And when I talked about how elephants can pick up vibrations through their feet and hear very low frequencies, hear or feel, however you want to say it, very low frequencies, they have equipment that can pick up those low frequencies, but we don't. So, can you advance to the next slide? Whoop, back one. Okay, so right here on this diagram, you see 1 wave goes from the middle to the top to the bottom and then back to the middle. And then a second wave goes from the middle to the top to the bottom and back to the middle. And that's happening in 1 second. 2 waves in 1 second is 2 hertz. 2 waves in 1 second is 2 hertz. So you're going to be asked about this on the exam, okay, about hertz and what they are, all right? You're going to have to understand this, and it's very important to understand you're talking about the number of waves per second, per 1 second. Okay, so how many waves happens in 1 second? If it's 2, that's 2 hertz. If it's 4,000, that's 4,000 hertz. Cool? Okay, good. But the other thing that we might want to know about sound is essentially how loud it is. And so how loud it is not about what the note is. You can play the same note, a middle C, you can play it loud or you can play it soft. That's about the loudness of the sound, or sometimes we call it the volume of the sound. And the way we measure that is not with frequency, but with amplitude. Okay, we call that amplitude. How loud is the sound? All right, how much energy does it have? So if we have a long wave, and another long wave, like we do at the top, those are the same note. Let's say that's middle C. But this one that has less amplitude is quieter. The less amplitude we have, the lower it goes up and the lower it dips down. Versus if the amplitude is higher, goes higher up and comes lower down, that would be louder. It has more amplitude. Whereas over here we have a deeper pitch. We have a deeper frequency. We have a lower note. It's a longer wavelength, and then this would be a shorter wavelength, which would be a higher pitch or a higher frequency. Okay? And again, we measure frequency in hertz. We measure amplitude in decibels. So you might see that, you have certain sound system that's capable of a certain number of decibels of sound. That means how loud can it go? And decibels are abbreviated dB. And it's actually lowercase d and then an uppercase b. So decibels is about how much energy there is to the frequency, how much energy there is to the sound. So you have both of those things. Frequency, what's the note? Loudness, that's the amplitude. Oh, so I have a little video, and this is just a real short video, and it shows what happens. What they did is they sprayed a little bit of water on a cymbal, and then they hit the cymbal and they slowed the video way down. And so what this is meant to illustrate is, in the same way that water comes off of the cymbal, can you play the video? Okay. In the same way that when the cymbal gets hit, like I say, it's in slow motion, this little spray of water comes off of that. If the water's not there, and even if the water is there, you're moving the air molecules in much the same way. Of course, the air molecules travel a lot further than water molecules do. But this is meant to illustrate how the air is being bent and changing the wavelengths of those vibrating molecules of air as they come towards your ear. Okay, that's all that is. Okay, so next I'm going to talk about the actual structure of the ear in the way that I began the vision lecture by talking about the structure of the eye. Okay, so I'm going to talk about the outside ear, the external ear, and then the middle ear. This is just about what happens to sound waves before you even transduce them. Okay, this is just about the sound getting into your ear. Okay, go ahead. All right, so here's what your ear looks like. And the thing that you probably think of as your ear is this flap of skin on the outside called the pinna. That's actually the least important part of your ear. Yes, it funnels sound into your ear canal, but that's about it. You can cut off your pinna, you know, Vincent van Gogh, right, was famously supposed to have done that, and you can still hear. There are many animals that don't have any pinna at all, and they can still hear just fine. You know, birds. Birds don't have any pinna. Well, then the next part of the external ear is what's called the external auditory canal. That's your ear hole. It's also called the external auditory meatus. You might see that. It's all just different words that different doctors have used for the same thing. And at the end of the ear canal is your eardrum. But the word that we use for that, for the eardrum in science, is your tympanic membrane. Tympanic membrane. Tympanum, or tympanum, they call it sometimes. Tympanum means drum. Okay, so it's the little drum inside your ear. And that's the thing that, you know, you can poke if you stick a Q-tip too far in your ear. So the eardrum, the tympanic membrane, the tympanum, is a little piece of skin that stretches across the inside aspect of the external auditory canal, and it covers it like the skin of a drum. And it behaves just like the skin of a drum. The air molecules that are vibrating come into your ear and they vibrate your eardrum. Behind that is your middle ear. And your middle ear, it's just filled with air, usually, unless you have water in your ear or something like that. Okay, so it's filled with, or an infection. It's filled with air, and it has 3 tiny bones inside of it that are called the ossicles. Ossicles means tiny bones. Okay, so the ossicles are the tiniest bones in your body. They're in your middle ear. If you ever have a chance to see actual ossicles, they're so cute and you should look at them. And the 3 ossicles are called the malleus, the incus, and the stapes. The malleus is also called the hammer. That's what malleus means in Latin, hammer. The incus is also called the anvil, and the stapes is also called the stirrup, and that's because of their shape. The malleus looks like a little hammer hitting on the incus, which is the anvil, and then that's attached to another little bone that looks like a little stirrup. If you ever get a chance to see one of those, I highly recommend it. They're adorable. So the vibrations of the tympanic membrane in turn vibrate the ossicles. And then the ossicles, the stirrup is the last of the ossicles, and it connects to the important part. In the same way that the light has to pass through the eye and get to the back where the retina is, the vibrations of the air have to pass through the auditory ear canal, vibrate the tympanum, vibrate the ossicles, and that's going to vibrate the cochlea. And the cochlea is for the auditory system what the retina is for the visual system. It's the place that's going to hold the receptors that do the transduction. Okay, so we're getting there. The place where the stirrup meets the cochlea, the place where it touches it, is called the oval window, and it's just because it's like a little oval-shaped window where that stapes is pressing on the cochlea to start vibrating it. Okay, so we're going to look inside the cochlea now. Okay. Oh, this is just another picture of the ossicles. That's all that is. Increased size. Next one. Okay. So now I'm going to talk about where the magic takes place. I'm going to talk about the inner ear. I'm going to talk about the cochlea and its different parts and what they do. This is what you need to know about auditory transduction. None of the rest of this was transduction. What was happening was stuff was getting vibrated by air vibrations. That's it. Now we're going to make that mean something. Okay, so down here in the bottom left-hand side is the cochlea. Okay, so the cochlea means snail, and it's called that because it looks like a snail shell. It's curled up and it's inside your ear. It's the inner ear, the main structure of the inner ear. And what we're going to do is we're going to take that. You can take the cochlea and it's coiled up, but you can take it, if you were dissecting the person's ear, you could take it out and you could unroll it and make it just like a long skinny tube. So what you see above that picture is a picture of a cross section of the cochlea. And the main structures that we're going to be talking about are in the middle chamber. And all of these chambers in the cochlea are actually filled with fluid. Okay? So they're filled with fluid so that the fluid could be full of ions. They're full of water, they're full of salt water, so that we can use those ions in order to do transduction, okay? So these are fluid-filled chambers, and when the air molecule vibrates the ossicles, it vibrates the cochlea, and the water molecules inside the cochlea start vibrating, okay? So we have vibrating fluid inside the cochlea, and those vibrations move these structures inside your cochlea in a very specific way. So there's the cross section of the chamber, and then this is the same thing just flipped over in the other direction. And then I'm taking down here that middle chamber and I'm blowing it way up. Okay, so here's the middle chamber and here's where the important stuff is. It's in this middle chamber. So the bottom of this middle chamber, the base of the middle chamber, this base right here is called the basilar membrane. That means the base membrane, the membrane at the bottom. Basilar membrane, like down in the basement, okay? This bottom membrane, the basilar membrane, is the one that holds the cells that do the transduction. Okay, so the base, the basilar membrane, holds certain cells in it that do transduction. At the top of those cells is another membrane. And that membrane is called the tectorial membrane. Tectorial means to touch. T-E-C-T, like tectum, touches the top of the tegmentum. The tectorial membrane touches the top of the receptor cells for auditory information. And those receptor cells are called hair cells. So the actual cells that do the transduction, they're right here, and they're called hair cells. And those hair cells are the ones that are responsible for the actual act of transduction. How does it happen? Okay, well here's what happens. So here's a picture of the little hair cells embedded in the basilar membrane, and here's the tectorial membrane touching at the top, okay? That's a picture of the electron microscope of the top of these hair cells. So here's the hair cells in a diagram with the tectorial membrane on top. That's the tops of the hair cells coming out of the basilar membrane, and they've peeled back the tectorial membrane so that you can see them. When the water molecules vibrate, when the liquid molecules, the fluid molecules vibrate inside the cochlea, the tectorial membrane moves across the top of the hair cells. And that is going to be the energy that we need for doing transduction. So the next slide. Okay, so here again, you see the hair cells embedded in the basilar membrane, the tectorial membrane touching at the top, and here's the tectorial membrane moving during the movement of the fluid inside the cochlea, which is caused by the vibration of the air molecules. One step back. So the hair cells have these little finger-like projections on the top of them, okay? So here's a hair cell, and it has these little finger-like projections at the top. Those little guys are called stereocilia. Stereo means for hearing, okay, it's for sound. Stereo is for sound. And cilia means little projections. Okay, so your fingers are like little cilia or like little hair-like things, okay? So that's actually where, it gets the name hair cell, from having these little projections at the top. And when the tectorial membrane rubs the top of the hair cells, they actually have little tiny gates on the top of the hair cells, and the action of the tectorial membrane pulls the gates open. So it's actually a mechanical process. The vibration of those fluid molecules causes the tectorial membrane to move, and it pulls open little gates. And guess what those gates are? They're ion channels. And so when the ion channels open, in this case potassium comes into the cell, and the potassium in the cell changes the amount of glutamate. Well, it's a little more complicated than that. You don't have to know all the steps of the process in between, but it changes the amount of glutamate that is being released by the hair cell. So hair cells are like photoreceptors. They work on graded potentials. They're always leaking glutamate, and when the potassium comes in, it changes the amount of glutamate that they're leaking. And then there are no significant intermediate cells here. The very next cell is the ganglion cell that receives the glutamate and will eventually go to action potential. So that process of just mechanically opening those little gates and letting the ions in is the process of changing physical energy, vibrations, into neural energy. Ions changing the amount of neurotransmitter that's released by the cell. So that is auditory transduction. That's how you hear. That's how you change the vibration of air molecules into the vibration of fluid molecules into neural energy. Okay. So can you go back one just real quick? So that cell, that ganglion cell that receives the information from the hair cell, that cell is going to, its axon is going to be your auditory nerve. The axon coming off of the ganglion cell that's receiving the information from the hair cells, that's your auditory nerve. That's the nerve that goes back to your brain, just like your optic nerve comes off of the back of your retina. Okay, so what happens next? Now we have a neural code, and now we have sent the information back towards the brain. Well, there are going to be a couple of stops. And one of the first stops is actually in the brain stem. And this is where we localize sound, okay? So it's there coming from the brain stem, sending the information up to the midbrain, and there we have the inferior and superior colliculus, and as I've mentioned several times, where the inferior and the superior colliculus meet, the inferior colliculus has the auditory information, the superior colliculus has the visual information, and that's where they get integrated, and so that's where we're able to localize sound. So information doesn't even have to get to the cortex. That's very early processing. Your ability to look and see where a sound is coming from. Okay, so from the inferior colliculus, the information is going to go to the thalamus. And the place on the thalamus, in vision, you'll remember it was the LGN, the lateral geniculate nucleus. In audition, in hearing, the place on the thalamus is the medial geniculate nucleus. So the medial geniculate nucleus is the auditory bump, the auditory structure on the thalamus. And then from there, from the inferior colliculi and the medial geniculate nucleus, then the information is going to go to your auditory cortex. Your auditory cortex is in your temporal lobes, and that's your primary auditory cortex. That's where you do your primary auditory processing. And then it goes out from there for more complex processing, and it gets shared with the rest of the brain. Okay. So now I'm going to talk about the actual process of what's going on inside the cochlea and how it can tell us about different pitches, about different frequencies of sound. Because it's actually done in two different ways because of the limitations of physical reality and the limitations of your brain. So your cochlea has actually two totally different kinds of processing depending on whether you're hearing something that's lower frequency or higher frequency. And then at the very end, I'm going to talk a little bit about hearing loss. Okay, go ahead. Next slide. All right, here we go. So here's the cochlea, and this is just a diagram. Yes, it represents, actual chambers, the actual chambers in your cochlea, and it represents the basilar membrane in your cochlea. And it's curled up like this, but obviously this is a diagram for purposes of trying to make this clear to you, okay? So imagine this is the coiled up cochlea. Imagine this is the little snail shell thing that's coiled up in your head. Over there on the left-hand side, we have the cochlea uncoiled, okay? So the setup for your cochlea is that the higher frequency sounds that you can hear, that processing is done at the base of your cochlea. And then the lower the frequency is, the higher up you have to go in the cochlea in order to do that processing. So the bottom of the cochlea is called the base, and that's where you do your high frequency auditory processing. And the very tippy top of the cochlea, like you're climbing up a mountain, because it's shaped like a little tiny mountain, like a little tiny snail shell, the tippy top of it is called the apex, and that's where you are able to perceive your lower frequency sound. So like I say, the beginning of human hearing starts around 100 to around 500 hertz, depending on how sensitive your equipment is. And then you can hear, some humans, very high frequency, especially when you're younger, up to 20,000 hertz down at the base. And so let's imagine we uncurl that cochlea like this. Down here would be the base, and up here would be the apex. Now here's the deal. High frequency information, high frequency sound waves are very, very fast, okay? 20,000 cycles per second is an incredibly fast oscillation of waves. Our brain on its own wouldn't be able to process that number of waves that quickly. But what it can do is create a cell that is affected by those higher frequencies. A cell that, when those high frequencies come through, it responds to high frequencies. And the way that it responds to high frequencies is that high frequencies actually the high frequency sound makes a peak in your basilar membrane. It makes the membrane peak in certain areas. And the hair cells that are located at the tops of the peaks are the ones that are going to be most stimulated. And so your brain knows if these cells right here at the tippy top, if they're stimulated, that's a high frequency sound. But the further and further down you get, if that part gets stimulated, gets pushed into a peak, it's a lower and lower and lower frequency sound. So that's how you're able to hear your higher frequency sounds 20,000 hertz is really high. You know, it's like the buzzing that you hear that comes off alarm systems sometimes, or you walk into the room, especially when you're young, you'll hear this really high-pitched ringing in your ears that's coming off of their alarm system, their security systems. There's tones that you can play on your phone that only young people can hear. They're at very high frequencies, and you play it in class, your older professors probably won't be able to hear it. So that's that kind of really high frequency. But around here, 7,000, 5,000, 4,000, 3,000, 2,000, those are still really high frequencies. You know, 7,000 cycles per second is still really fast, but that's where human speech is. So the vast majority of your hearing is dedicated to the frequencies that human speech is in. Okay, that's what your cochlea wants to do. Then what about these lower frequency sounds, like a bass note, or you might hear a low pitched boom in the distance? You can still hear those low frequency sounds, but the problem with low frequency sounds is that the wavelength is low frequency. A wavelength that's low frequency cannot make a peak in the basilar membrane. The frequency is too low to create a peak. And so you cannot hear low frequencies using the same system as you use for high frequencies. It doesn't make a peak in the basilar membrane. So your body developed a different way of transducing low frequencies, which is what you do at the apex of your cochlea. Well, remember how I said before, high frequency sounds are so fast of wavelengths that your brain can't count them? That your brain can't count that fast, that many cycles, that fast? Your brain can count 200 or 300 or 100 cycles per second. That is slow enough for the capacity of the human brain. So at the apex, your brain actually counts the number of cycles per second, 100, 200, 300, and it has the capacity to do that. So that's how your brain hears low frequency sounds, just by counting the number of cycles per second that are occurring at the apex. So you have those two different systems because you need to have those two different systems. Oh, and the one thing I want to say about that is those two kinds of coding, those two kinds of neural coding have different names in perception, in perceptual science. The higher frequency coding that we have makes a peak on the basilar membrane in a certain specific place. And so that's called anatomical coding. Anatomical coding. Anatomical coding is about what part of your anatomy is being stimulated. Is it this very specific part of the basilar membrane anatomy, or is this other very specific part of the basilar membrane anatomy that's being touched, that's being affected? Okay, so that's anatomical coding. The other kind of coding where you count the number of cycles per second, the one for low frequency that happens at the apex, that one's called temporal coding because it's about time-based information. How many cycles per second? It's time-based information. Temporal coding. Okay, so you use those two different ways. Okay, so now I'm going to talk a little bit about hearing loss. You can have different kinds of hearing loss. You can have peripheral hearing loss, where you have damage to the external ear or the middle ear, or you can have central hearing loss, where you have damage to the inner ear, where you actually have damage to the cochlea or the auditory nerve. Okay, so peripheral damage can happen in a number of ways. So this is, the ow, you stuck the Q-tip through your tympanic membrane. And, if you're young and healthy, it's going to heal. It's going to heal pretty quickly if you rupture that eardrum. Yes, everything will sound tinny for a while, but the air will still travel. It'll still vibrate the ossicles, and it will still eventually get to the cochlea. It'll just sound a little off, like you're hearing in a tin can. That's a kind of peripheral hearing loss. You might have people who the middle ear gets lots of infections when they're little, and it creates a lot of scar tissue, and then you don't have good vibration of the ossicles, and so you don't have good vibration of the cochlea. So that can affect hearing. And that's why we like put tubes in kids' ears when they're little, because we don't want them to develop that scar tissue and have that peripheral hearing loss, because it is, annoying and can make it so that you don't hear as well. But what happens when we actually have damage inside the cochlea? What happens if your hair cells, usually this is congenital, okay? Usually it's something that happens, it can happen to somebody when they're just born with damaged hair cells, but then there's other things that can damage hair cells over time too. Let's say that you have damage to your hair cells, or you have missing hair cells, okay? What can we do? Well, somebody was listening to their boring psychology auditory lectures, right? Some fun person was listening to this lecture, and they figured out that the different frequencies of sound stimulate different parts of the cochlea, stimulate the different parts of the basilar membrane on the cochlea, from the base going all the way up to the apex. And they looked and they saw the auditory nerve, and if the auditory nerve is fine, what would happen if you just stimulated the auditory nerve in those places? Would the person hear a sound? And the answer is yes. You can electrically stimulate those ends of the auditory nerves in those specific places, and the person will hear that as a sound. So what we've done is we've created something called a cochlear implant. And a cochlear implant has a microphone on the outside of your head that picks up sound, okay? And the microphone attaches to an implant on the inside of the head that has the little electrical stimulators in it to stimulate all of the places of those nerve endings along the basilar membrane, all those auditory nerve endings. So when different sounds come into the microphone, it activates the different little electrodes to essentially give a tiny, tiny, tiny little shock to the ends of those auditory nerves. And so the person can hear. We found a way to bypass the hair cell situation. Because here's the thing, most hearing loss that is acquired, most hearing loss period is acquired as you go on in your life and loud sounds start to make you deaf. So loud sounds damage your hair cells. And what it does is loud sounds, very high amplitude sounds, very high volume sounds, make a lot of action on the basilar membrane. It really vibrates the crap out of those little hair cells. And the little tops of the hair cells will get damaged over time. Can you go to the next one? Okay, so here's a picture of intact stereocilia coming out of the top of hair cells. And here's a picture of hair cells that have been blasted with high volume, high amplitude sound. The stereocilia get damaged, it breaks the hair cells, they get over flooded with ions, and they die. So when you're listening to very high volume sound, it is creating a lot of vibration, and the part that gets most impacted is the base of the cochlea, where your high frequency sounds are transduced. Those are going to be the ones that get the most action and the ones that get broken first. So you won't be able to hear high-pitched sounds. And the problem with that is that the consonants in human language are high-pitched sounds. So you're going to lose your high-pitched frequency hearing first when you listen to music that's too loud. And you're not going to be able to hear the consonants when people speak. The consonants are the P's, the B's, the M's, the L's, the N's, the T's, the K's, okay, the S's. All of those things, consonants, you can't hear those, and so the only thing you can hear that people produce are the low frequency sounds. And those are the vowels, the E's, the Oo's, the Ah's. And so when you have that kind of hearing damage, everybody sounds like Charlie Brown teachers, Charlie Brown adults. Have you ever seen Charlie Brown Peanuts, cartoon? The adults go, wah, 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 wah. That's what it sounds like to have high frequency hearing loss, acquired hearing loss from listening to music too loud. So when you hear the person roll up next to you and she's playing her music really, really loud, you hear the bass. The bass is low frequency, and low frequency can travel through solid objects, through liquid, through air very easily, and come into your car, so you hear that boom, boom, boom. That's the bass. But that's actually the part of those are the frequencies that are the least likely to damage your ears. Inside the car, where Mrs. Cool Stuff right there, she hears the treble. She hears the top registers. She hears not just the bass notes, she hears the higher frequency notes, and that's damaging the hell out of her hearing. Okay, so not a good idea to listen to things really, really loudly. Use your earplugs. Listen to things at a moderate volume. Don't shoot off guns next to your ear if you can possibly avoid it, or loud things like that. It does damage, and then the only thing that we can do at this point is if you want to go through a cochlear implant to get that kind of issue fixed. I do want to mention that research is now looking at fixing hair cells. We're trying to see if we can use stem cell implants to fix hair cells. And some of the first experiments we did were in birds, and it was very promising. We were able to implant stem cells into chickens and other birds and regrow their hair cells after damage. Repair the stereocilia, regrow the hair cell, repair it. But we haven't been able to do it in mammals yet. When they tried it on rats and other mammals, it didn't work. And we're not sure why, but it's a growing area of research, seeing if we can use stem cells to repair that central hearing loss. Okay, so that's it about audition. The next lecture you want to listen to is the one that's about gustation and olfaction. Okay, I'll see you in class.